it's not about, I mean, we could have easily dug in our ditches and shot at each other and saying, like, my method is better, like scientists usually do.
And then there's a kind of rare collaboration like the one that Jörn and I have, which for me has been transformative for my lab. And there's something deeper going on, and that is really hard to come by. And it's very interesting to think about what's required for that to work out.
How things are arranged on the cortical sheet is probably not that important for the computation.
You could argue either way. I think, these things are very replicable and they are true. And I think the placement of components in the brain is very important.
The key lesson from the journey, I think, is that
this is Brain Inspired.
Hey everyone, thanks for listening. I'm Paul Middlebrooks. So on the one hand, you have brains. One way to think of brains is as a series of encoding and decoding information processing steps. Sensory areas encode incoming signals from the environment. Like say you see a friendly acquaintance and he says, "Dude, I saw the movie Inception. Have you seen it? You're a neuroscientist. You would love it. Is that how dreams really work?" And this auditory signal gets encoded in some parts of your brain, but then you could say to pass it along to the next part of your brain and ensure that it maintains its representation as the same auditory signal, that encoded information needs to get decoded so the next brain area knows that it has an incoming auditory signal. And on, encoding and decoding, until there's a final decoding step that results in motor output. In our example, that would be silently turning around and walking away. On the other hand, you have neuroscientists. Neuroscientists have tools to measure brain activity and ask how that information gets represented in brains. And these days, we have big brain data, big data from brains. We measure lots of brain signals from lots of different brain areas in parallel. So where do we begin with that? What's the right level to ask about? Does it matter what parts of the brain a signal comes from? Can we ignore that location information and focus instead on how the brain activity patterns differ among different stimuli? Or when does it make sense to focus on some bit of brain activity in particular and ask whether it can tell you if a person is about to engage you in some awkward situation or not? Jörn Diedrichsen and Niko Kriegeskorte, my guests today, have called these different layers, these different ways of examining what's represented in brain signals, the onion of brain representation. The idea is that as you peel the onion, you remove some facet of information that may or may not be relevant to your particular goal of understanding. And that's, of course, a cursory and insufficient introduction to their review paper that we're talking about, which is called Peeling the Onion of Brain Representations. So Niko has been on the show before. He's a professor at Columbia, and he studies visual perception and how we can use deep learning to better understand how brains visually perceive the world. And Jörn, who's new to the show today, specializes on the motor side of things. So he runs his lab at the University of Western Ontario, where he studies the learning and production of coordinated and skilled movements. Jörn and Niko have been collaborating for a few years now on representational models of brain activity. And because of that, and because they're joint authors of the paper that we discuss, I decided to try having them both on at once, which presented a few challenges, but I think it turned out well. I enjoyed it. You may remember last episode I had Michael Rescorla on, and Michael and I talked about representation in philosophy and in brain and machine computation. And today I got Niko and Jörn's view on representation as well. Then we go on to talk about the onion, peeling it and putting it back together, and we cover plenty of surrounding topics as usual. Patreon supporters, I'm working on getting a separate podcast feed going, especially for you, so I'll announce that when it happens. It'll have extra bits of interviews that I will save just for your personal podcast feed as a way of saying thanks to you. So that will be coming soon. And in the next month or so, you'll be getting parts of the Neuroscience AI course as it comes out. Bolaji, Jörn, Sebastian, Jan, Jimmy, and Michael, thank you for your recent support on Patreon. Show notes for this episode are at braininspired.co/podcast/61. 61? Okay, here's Jörn and Niko. So I'm peeling the onion of brain representations, and I'm having this moment where I'm reading the text, and the text is referring to one of the images, and I'm looking at the image, I'm looking at the panel in the image, and it's not like matching up for me. I'm like having a hard time like understanding how they relate. I keep going back and forth. I'm getting kind of frustrated, but then it kind of starts to make sense, and I'm starting to kind of get it, and this kind of goes on. And I finally convinced myself, oh, okay, yeah, now I understand how the figure and text match. And I realize finally that I'm actually looking at the wrong figure. And, the scary thing is I, halfway convinced myself that I was, that it all makes sense, right? And this is not a good sign for a scientist, for things like confirmation bias and et cetera in my own practice, right? But I also recognize this thing has recurred in my life, reading papers, and it's like, what the hell's wrong with me? So how can I do science if I can't match figures to text?
Jörn, we failed.
We failed.
It's me. It's my failure as the reader.
No, your failure is our failure.
It actually made complete sense once I matched the text to the correct figure. So
okay, good.
Jörn, welcome to the show, and Niko, welcome back. Thanks, guys, for being here.
Good to be here. Good to see you.
Good to see you too. All right, so just to kind of start off, I mean, we all have these hurdles to overcome. This sort of resistance. Mine, obviously, is just a general incompetence. But, for fun, I thought I might start by asking both of you, maybe for an example of some hurdle or some challenge that you might face, either these days or maybe something that you even overcame earlier in your scientific careers, in your scientific journey. Jörn, you're new. I'll start with you. Is there anything that you can think of?
I mean, I have one thing that relates very highly to also writing this paper, and maybe even the problem you had in matching text to figure, is that things can be extremely clear in your brain and in your mind, and you kind of think you perfectly understand what you're talking about, but actually the misconception that just saying the words and then expecting other people to know exactly what you mean is a very treacherous trap to fall in. Yeah. Okay, and so what I really learned is that you cannot be clear enough, and even if you're the person that you talk to nods and smiles at you and says, "I perfectly understand what you're saying," you cannot be sure that they actually understand really what you're saying.
As the podcast host nods and smiles as he says this.
And if I have learned one thing, is that I can work for hours and hours and hours in trying to write something that I think is crystal clear. And then when I get the reviewer or comments back, they say that I didn't understand it, or they completely misunderstand it, and they are very sure about that they understood it correctly.
Yeah.
So for all my trainees, that's the first thing, and every trainee falls into the gap. They think immediately what they say is understanding, because obviously they talking about the experiment and the supervisor knows what they're talking about, but actually getting the right words, making it very clear, and making sure that the representations in the other person's brain are actually the representations that you want them to be, nice. Is not a very easy feat. And that's, so I'm still failing on that every day, but, at least I'm warned that is a big problem.
It is, right? Writing is really hard. Any type of writing.
Or even talking.
I wouldn't know about that. Yeah.
In a direct conversation, you at least have the option to question and to talk and to make sure that it's coming across right. Yeah.
This is a challenge, ever going challenge in a podcast format as well, which it's weird because I expect a lot from my listeners, and it's always hard to know where the right balance is, and who knows if I strike it right. Some people seem to like it. Some people, I get all sorts of comments like, it was over my head, but I enjoyed it. And I think, I'm not doing my job, Anyway, so Niko, how about you? Are you perfect?
My life is a series of failures, and I constantly feel inadequate, and it's always been like that, and insecure about, what I understand and what I don't understand. And of course, I mean, as everyone always says, we learn from error, right? So this is what drives us to improve things. That's just a sense of how we're not quite there yet, right? And so it's a good thing, but it's definitely extremely uncomfortable. So a lot of my life is spent in relative discomfort. And when you see people give talks, then that's very misleading, because of course when we give a talk or we write a paper, that's after years of thinking about this stuff, right? And then we leave out all the things that we're not confident about. We just say like the little sort of pearls of insight that we've arrived at. And then we seem very confident and in control, but that's just because we choose to only move in the small part of this whole space in which we are comfortable and confident.
The lowest error in the error surface is what we present. There's this meta aspect to this too, though. You have to sort of be comfortable being uncomfortable and just get used to it. It's a strange thing, but useful.
Yeah, this reminds me of a song. I think Fiona Apple. I'm good at being uncomfortable, so I can't help changing all the time. I felt like, that's
nice.
That's a good description.
That's good. Yeah. I also tell my students that failing should be fun, and all you need to do in your PhD is the trick is to really fail very fast, right? And just not. Try things out, fail, learn from it, move on. And that iteration is really important, I think. And then knowing that that's part of the process, that's really important.
Yeah, the resilience that comes with that. This is all really good stuff. So, all right, very good. I guess none of us are perfect. Right? This shatters my preconceived notions here. How did your collaboration come about? Did you guys bond over your hatred for neuroanatomical spatial locations, or what? How'd you guys come together on this recent paper, and then you've written a couple before as well?
I think we met. How do we meet? I think you contacted me because you said, I read your paper. I thought that was interesting. The original. So
I remember two things, and I don't remember in which order they happened. One was that I reviewed a paper of Jörn's, a very important one that is sort of the basis for our collaboration since, which is the paper about pattern component modeling. Mm-hmm. And I was very inspired by that paper, and also awed a little bit. And I was struggling to follow the math, but I could tell that this is an important thing, and that it has some elements that are missing from my framework up to that point for analyzing brain activity patterns. So it was really an inspiration, and I knew like this guy is onto something, and this guy has something that I need, and so that was like one step. And then the other thing was that I think we wanted a little bit, maybe then I talked to my graduate student Alex Walter, and we wanted to play around with Jörn's methods, and I think we contacted him about the code for some piece of his analysis, and he shared that, and that began a conversation where in the end my graduate student moved to Jörn's lab for an extended period, and we started collaborating, and we haven't stopped collaborating ever since.
Yeah, and in the from my perspective was, he commented on my paper, and then I realized that the problem is, so PCM and representational similarity analysis are very related methods, and with PCM that might be elegant, the math might be compelling, but obviously it has a lot of problems for people to use it, to be intuitive, and all those things are given in RSA. So there were really a lot of things that are important about statistical development that really RSA had nailed. Namely intuitive, the option to explore the data, data visualization, and so on. And so, despite the fact that we had these method developments in my lab, we actually used RSA very heavily. And I think it's been a great conversation. And what I really value is that ultimately it's not about, I mean, we could have easily dug in our ditches and shot at each other and saying, like, my method is better, like scientists usually do. But the awesome thing, I think, about the conversation is that we both, I think, Niko, and I hope I also understand that methods are just tools. And the key thing is, like, the question is what you answer with them. And that's what we feel passionate about. And with the tools, you just choose the best one and the most appropriate one. And I think that's been like the underlying current in our collaborations that I really value.
Well, I was going to ask about collaborations next, just in general, because if you're not an important university unless you tout your interdisciplinary nature and that you foster collaborations between departments and stuff. And so how important do you think is collaborating versus really digging in your heels and maybe not competing against each other, but sort of focusing on your own narrow pathway? What's the right balance there?
I don't know. I think it's a little bit like, this might be a slightly hokey analogy, but like bike racing, right? So bike racing is a sport where you need to, do your own thing and you need to work hard. But, if you don't work together with other people and form those coalitions within the race and talk and, get into one common goal, then you're never going to succeed.
You're leaving out the performance-enhancing drugs, but okay.
Oh, there's that. So I've skipped those. So I'm talking about clean bike racing.
Yeah.
Here. But yeah. So, if you piss people off and you're an asshole, then that's the end of your career, usually. Well, doesn't count for all of the people, but that's certainly the case. And that's also in science. I mean, there's an interesting, sometimes you need to stand up for yourself and say, this is an idea that I've worked on, and here's my work, and not basically put your light under a shovel. And that is unfortunately often what happens with some trainees, often female, but also for some male trainees. It's not natural. The really good and very smart people sometimes don't have the urge to really go out and bang their drum. And there are other people who bang the drum very well, but fall short in some other departments. And both of them are skills that you kind of, I think, have to have. But in terms of collaborations, I mean, it's also, we're spending a lot of time on science and we need to feel passionate about it. And that also means in collaborations, the personal level is also really important. I mean, you really need to get to like the other person, right?
Yeah.
And not just for their skills, but also for their opinions and general outlook on life.
You have to respect them anyway. I know in the decision-making world where I come from, people are kind of entrenched in their camps. And looking at it from the outside, you think that everyone hates each other. But in reality, everyone's going for this common goal, and they do respect each other, even if they do, have plenty of friction between them. But your collaboration between Jörn, you and Niko is really not frictional at all, right?
Oh, I wouldn't say that.
Oh, well, so what is something that you guys disagree about besides?
Oh, everything.
No,
what do we not disagree about? No,
we have fundamental disagreements, for example, about the naming conventions for functions in our new Python toolbox and stuff like that.
Important stuff.
So, it is hard. It is hard. I have to say, it's very tough. With Jörn.
And we're both very stubborn, so that doesn't make things easy.
Yeah, he's obstinate. He doesn't want to understand.
But we also fundamentally disagreed about this onion paper.
Oh, you did?
Yes, absolutely. I hope it shows in the writing.
No, I don't think it does. Is it too soon to talk about that? Should we get into the details before?
Maybe one thing about collaborations. I think there's kind of very qualitatively different modes of collaboration, right? There's sort of just positive interactions with other people in the field that can be critical but still lead to something good. Then there's the kind of collaboration where you have something, like you've built a tool and you share it with others and maybe you give advice or something like that, but it's somewhat asymmetrical.
Yeah.
And then there's a kind of rare collaboration like the one that Jörn and I have, which for me has been transformative for my lab And there's something deeper going on, and that is really hard to come by. And it's very interesting to think about what's required for that to work out. And I think it has to be sort of this element of complementarity, where there is a common goal. And in our case, maybe that has to do with wanting to measure brain activity patterns with massively parallel methods, such as functional imaging and cell recordings, and then linking these measurements to theories, to be able to test theories. But then there's also sort of a complementarity where Jörn works in the motor domain, and he's interested in motor cortex and motor control. And when he talks, he uses, metaphors from biking, which I would never come up with. I don't know. When I see a biking race, I see only competition. I wouldn't even, notice that there's, this is a very subtle thing that I'm hearing about for the first time. So it's quite inspiring. I'll watch the next, differently the next time I see, some bikers flash by on TV. So, and I am focused on vision and on perception. So there's a fundamental complementarity there. And then also in terms of intellectual styles, there's a complementarity, right? So I feel like deeply that Jörn has something where I have shortcomings, right? So he has some abilities that I feel that's exactly what is sort of missing, what helps me solve my problems. And I just want him to help me solve my problems. And I can also learn a lot, right? So because I love the things that he's good at, and I'm kind of suffering from the fact that I'm not as good at them as he is, but this inspires me a lot. And I'm also, I know that I can make progress there. I know that it's not a loss. I'm not a lost cause. I can always learn more. And, for me, that's how it works.
Yeah, I can return the compliment. I mean, I feel with other things, especially the same way, that there are things that I just don't, Niko, it's often like it takes all kinds in science, right? I mean, there's, we can't just say, oh, I'm not as good as that person, or I'm not as famous as Niko Kriegeskorte. And you can feel that, but ultimately the key thing is you need to know what you are and be proud of it, and then respect other people for what their strengths are and what they bring to the table. And yeah, I mean, there's just so much that I've learned from Niko that is fantastic.
Is this collaboration style, is this the modern version of the Renaissance person, I'll say, to be politically correct, where, back in the day, there wasn't as much to know. I mean, there's still a lot to know, but someone could sort of touch a lot of different areas. But now everyone's so specialized that everyone fills each other's holes. Are the days of the lone researcher gone?
I think the days of the lone researcher are gone. Yeah, I think if they ever existed,
if they ever really existed,
if they ever really existed. I mean, I doubted that they ever really existed. Nobody came up with ideas in vacuum. It's always based on communication. And when you think
but
about
there are people
places,
like Newton, who worked in his study, doing his alchemy.
Yes and no, but he was embedded in the natural philosophy movement in Britain at that time, and there was a lot of people that discussed and wrote and interacted. And without that community, Newton wouldn't be Newton. So there is a reason why there are hotspots of high science coming up, and it's not the lone wolf that suddenly comes up with this smashing theory. Although, history then post hoc constructs it because we want heroes and single people that do these things. But, that's never how science was done. But I think, moving forward, I think, yes. For me, it's been transformational here at Western to have my lab not alone, but also here together with two other PIs and co-supervising. And it's transformational to have Niko as a collaborator and send students back and forth and exchange ideas and so on. I mean, they change the way you work, and they change the way you think about science and how you value things. And they also can change the way that your students interact with each other and get to know science.
So Niko, you pointed out that you are more concerned with the perception side of things, and Jörn is more concerned with the motor side of things. But you're both on board with this concept, I think, that I love, that movement is the primary goal of brains. This Daniel Wolpert approach here. And, there's this idea that actually thoughts are sort of modified motor commands that have been internalized throughout evolution. And in that sense, thought is a sort of proto-form of action. So am I right in thinking that you're both on board with this movement is the goal of brains, essentially? And if so, is this the lens through which you view all of your brain research, whether it's motor or perception?
You need to ask Niko about that one.
So I first heard this argument from my colleague in Cambridge and now colleague here, Daniel Wolpert,
yep,
whom I greatly respect. And when I heard him say that, I found it entirely compelling. And I still find it quite compelling, but there's also sort of a part of me that wants to question it and argue. And that's because the perception to action cycle is a cycle, right? So you could also call it the action to perception cycle. It depends how you sort of, where you draw the line, where how you sequentialize it to think about it. We have a focus on the brain, and perception is the input and action is the output. But there is an arbitrariness there, and there's a very, very interesting philosophical question, I think. But I think it's sort of maybe too big for right now. But I would love to, explore this more deeply, because it is a deep question. And you could say, we want to control our actions, but just so that we can fulfill our needs and that we can perceive the world. What the ultimate goal is sort of in the eye of the beholder, right? Ultimately, there might not be any ultimate goal.
Oh man!
Yeah, I think that this idea of movement first is really born a little bit out of a minority complex that the Mutter community has. I mean, look, if you look at neuroscience, there's the ratio of vision research to motor research is maybe 8-1 or something,
right.
In terms of mass.
And that's just vision. That's not even just all of perception.
Vision, yeah. And so you kind of, when you see that and you kind of think about, would I rather lose my sight or would I rather be quadriplegic? It's certainly not an 8-1 decision clearly here, right? So, but I think, the interesting thing is that it gives you two very different perspectives on representation and how you think about representation. And I think the way that the old representationists have thought about representation is really just basically driven by perception, right? And when we need to represent these objects in the world, and we need to have codes that stand for these objects. And I think that idea is not coming as natural when you think about controlling.
Right.
So I think that's kind of an interesting tension field. And ultimately, whatever theory or whatever concept or framework we have for representation, it needs to capture both of those sides.
Well, let's go ahead and talk about representation models. So, and to kind of start off, let's keep kicking around the idea of representation itself for a minute. I just had philosopher Michael Rescorla on, which you guys wouldn't have been able to hear. It hasn't been released yet, anyway. But we went kind of deep on the history and the current landscape of representation in philosophy, in particular, in philosophy of mind and the philosophy of computation.
Mm-hmm.
And what's relevant for today is that he distinguishes between something that you're trying to understand, namely like the brain and mental computation, the mind, and how representation is the right way to understand computation in brains. This is called the representational level in computational field, versus something that you're building, which, a computing machine, and how for that, all you really need are formal rules and properties. You don't need to understand what will be represented by those rules when you're building the computing machine. And this is called the syntactic level in the computational work. But neural networks, there's this, weird in between, right? Because yes, we do build them, but with as few formal computational rules as possible. There are constraints, right? So we build them, but we're also trying to understand them as computing systems, and in the hopes to better understand our brains and our minds and how our minds compute. I don't know. With that in the background, sort of, how do you, think of representation in computation in general and its relation to neural activity in particular? And I don't know who might want to jump in.
Maybe I would ever so slightly disagree with the idea that computer scientists can do without the concept of representation. I think computer scientists, even when they're not working with neural networks, but certainly when they are working with neural networks, think of the states of the system as representing things like numbers.
So in a simple Turing machine, you think this is the case?
Yeah, the memory cells represent things. I think that is already a deeply representational way of envisioning a computer. And as for all the computation models that I'm aware of, I feel like that's the case. But it's a less problematic way of thinking about representation because these are systems that we build ourselves. So it's much more crisp in terms of there being a single sort of abstract description level that involves representation that perfectly explains how the system behaves. And things become more murky when we use that abstract language of description to understand a natural system like a biological brain.
An abstract syntactical type level, like non-representational level, you mean?
No, an abstract representational level. So the language that's natural for computer science is also representational. And then we want to apply that to the brain. And I think that is well motivated. It's our best shot. But it's also problematic.
Yeah, I would agree. I mean, so I'm not quite sure when somebody says I'm a non-representational view of something. Now, you can have a non-representational view of a system itself, but what you can't have is a non-representational view of the relationship between a system and the world. So a computational system and the things that it relates to, that it computes, that it acts in. That relationship between the brain and the world, that is a core definition of representation. How does things that happen in the brain relate to things that happen in the world?
So this is your definition of representation. So there's nothing...
Right, and this is like a super, basically, umbrella that is, in this sense, void of meaning. But all I'm saying here is that I'm not quite sure if I understand when people say, so if you take a non-representational view of the brain, you can only talk about the brain in resting state and look at these fluctuations. That's a clear non-representational view. But as soon as you talk about how does this neural activity relate to anything that the brain does or sees or experiences or tries to achieve, you're talking about the relationship between things that happen in the world and things that happen in the brain. And that relationship, that's the philosophical core of the term representation for me. Now, yeah.
Well, I was going to say this is intentionality or aboutness.
Intentionality, aboutness, exactly John Searle's intentionality and aboutness. So now there are like two radical views, I think, on this. And I think the paper is trying to kind of get right in between and kind of break up the front lines. So on the one hand, I think that what we would call the old representationalists in neuroscience, right? That say like, okay, here's a neuron, I measure it, and it clearly represents velocity of the arm, or it represents the color of the object, or it's a Gabor function, okay? So that basically what happens here is there's a one-to-one relationship between the information contained in the firing rate of the neuron and some physical variable that we use to think about the world, some feature.
Grandmother cell.
Yeah, yes, but also it doesn't have to be. It can be distributed code like population vector in motor cortex is a classical example, where we say like, oh, yes, there's movement, direction, and velocity. That's a Newtonian concept about how we describe arm movement. And there's cell activity, and we just say, yeah, that's exactly, there are these cosine tuning functions, and we're done. Now, that's what we've called the feature fallacy in representational modeling, that you think that the way we think about the brain and break it up in physical variables, that the neurons encode it in the same way and are well described by these features. And I think in the visual cortex, that has given rise to a Nobel Prize, Hugo and Wiesel, right? Now in the motor cortex, they had, I think, you can sum up the motor cortical neurophysiology field for the last 40 years by having really Hubel and Wiesel envy and trying to find what is the code in motor cortex. And they, paper after paper after paper is the idea that, it codes a little bit for velocity, it codes a little bit for muscles, it codes a little bit for something else, and there's a lot of mixed selectivity, and we don't actually know what's going on.
Mm-hmm.
So most papers are like this. And so I think that triggers the other side to say, let's throw out the baby with the bathwater and not think about representation at all. And it's all the dynamical systems, and there's some rules, and then now you might call those people that say, I don't want to talk about representation. Now, the interesting thing is that if they don't want to talk about representation, they can't actually talk about how those dynamical systems relate to things that the brain wants to do, like moving their arm.
The dynamical system has to produce the thing, right?
Well, the state of the dynamical system relates in some lawful way to the things that it needs to produce. Okay? And not just what it produced, but the internal states, in a way they relate to things that are context, what is the next movement or whatever. And I think there is no intellectual distinction, no clear intellectual distinction that shouldn't be called representation. So I call that representation because it's about the relatedness of things that happen in the world and things that happen in the brain. And studying that relationship, I think, is very important. So when we study these recurrent neural networks or, dynamical systems, we can think about attractor points and oscillations and, beta waves. But ultimately, we also need to think about how does the difference in the states relate to the things that the thing wants to do or sees or represents.
But is that dependent on your interpretation of the system, or is it inherent to, let's say, the dynamical system, right? So are you adding anything by saying that there is representation, or is it inherent to the system?
Well,
yeah, that's a great question. So my view is that representation gives us a more abstract language of description on top of dynamical systems. So it's not incomplete to say it's a dynamical system and we can study it as just a dynamical system and never refer to representations. We can do that, but the representational interpretation is powerful in linking what goes on in the brain to the world, as Jörn said. And this gives us a functional understanding, much like in biology, when we want to understand organisms, we understand the functions of different parts of their body. What's the function of a hand, right? And we can come up with all kinds of stories, and that is, on the one hand, somewhat arbitrary. There's the problem of just so stories in these interpretations of how a body of an animal might be adapted to its environment, but it's also clearly a very powerful explanatory concept, right? And representation, to interpret something as a representation, is to ascribe a particular function to a pattern of activity in the brain. And the function is to convey particular information about the outside world. And that requires that the information be present there and that it's encoded in a format that can be exploited by the rest of the brain. And it also requires that it actually is exploited by the rest of the brain, right? These three criteria.
So this is a terrible question, but the analogy of a tree falling in the forest when no one is around to hear it just popped into my head, right? So the tree has the ability to produce sound. It's all there in this really terrible metaphor, so I apologize. But if there's no one there to hear it, then one might say that, would one say that the sound still happened? So if there's no one there to interpret that this is a representation, is it a representation if there's no mind to interpret it? And we'll move on, I promise, after this.
Yeah, I mean, the answer to that is if nobody's around to see the tree falling, who cares?
But there's a model in a forest, and it is no. Okay, so we'll go with who cares.
Well, no, I mean, this is important, right? So the tree is still falling, but the pattern of activity that is not read out, that can never, even in principle, affect behavior in any way, is not a representation.
But I mean, to answer your question, like, are representations real or not? Well, nothing is real, right? I mean, none of our models, the brain is also not a dynamical system, right? At some level of description, a dynamical system is a good analogy or powerful model about how the brain activity might evolve on the short-term scale. But it's not a dynamical system. It is not a dynamical system. And likewise, the brain does not have representations. They do not exist. But it's a conceptual tool to understand the relationship of neural activity and things that happen in the world.
Niko, are you happy with that?
Almost.
Okay.
For me, there's slightly more arbitrariness at the level of representational interpretations. So my conception is a little bit lighter weight. So I've been talking regularly to a philosopher at Barnard College at Columbia, John Morrison, and he has a slightly stronger representational view where he thinks, so more of an ontological interpretation of representation, where he thinks there are systems that really do have representations, and there are other things that really don't have representations. And I'm not comfortable with that. And he gets there by much the same argument that Jörn kind of started, although I think he wouldn't pursue it to this consequence, which is that for all the levels equally, it's questionable how exactly they relate to the reality, right? So it's unclear why we should make weaker claims about this particular level of description that we call representational. But to me, it seems that the arbitrariness that I experience when I try to pull it off and trying to understand how the brain works is at a level where I am closer to Dennett in philosophy, which John tells me is a minority view in philosophy, in saying that we take the intentional stance and we describe the system as though it were representational. And maybe in some deep sense it is, but there's not only one representational interpretation. And you could apply this language to any system, even to a thermostat, except when the system is very simple, then maybe you have no need for the heavy conceptual artillery of representational language to kind of make sense of what the system does.
I suppose, Jörn, you don't think representation is computationally causal then, and Niko, you probably don't either. You can't do computations with representations, right?
Yes, you can.
Oh. You can.
Only.
You can only compute with. Oh, I see, because it's embedded within the structure of the computer.
Computations are performed on representations, I would say.
There's no separation between the symbol and its meaning.
These go together.
Well, yeah. Computation is performed on representations. So once you are at a representational level of description, these representations are causal. You take that representation away, the computation doesn't work anymore. Now, once you're at a level of description below it, meaning you look at the electron flow in transistors or in spiking potentials in neurons or membrane potentials, then you can choose not to talk about representation and the system still works. So there it's non-causal. So it's really confusion about levels of description, I think, here. And the key thing that is a rather simple idea is that as you move up in the level of description, the concepts that you need to build causal models become different. And there is a non-trivial relationship between them. And physicists have understood that for a long time. And I think neuroscience is just catching up on that.
Yeah, it's really, whether it's causal or not, at many different levels of abstraction, you could have a causal model, right? And there is not greater causality at lower levels of abstraction. In fact, the dynamical systems perspective is non-causal, right? So you could even argue it's the other way around. At this higher level of description, where we think about representations causing downstream other representations through computations that transform them, that is more of a causal interpretation. than a dynamical systems description, which is mathematically defined, but not mechanistic in the sense of cause and effect.
All right, so I think it's time for us all to take a shot of our liquor, and I think that's actually a good transition to talk about encoding and decoding to get into the onion. Quote of the show so far, by the way, is Jörn's, "Nothing is real." I believe that's a direct quote. That's good. So representations and encoding and decoding, the brain has to encode and decode. And so encoding is basically how a stimulus gets translated into neural activity, into some, let's say, representation. Whereas decoding, which is, so decoding is talked about in two different ways. And the way that you guys use it in the paper, and I think properly, is that decoding in terms of the way that brains function is translated some encoded neural activity into something useful for downstream neural activity and or motor output or behavior. And brain processing is one big cascade of encoding and decoding until a behavior is produced. So it's this interesting, you don't often think of it, but one area in the brain has to be an encoder and a decoder in that respect. Do I have that picture right?
So these are confusing concepts, right? I remember when I was an undergraduate, we learned about the brain, how signals travel from the senses to different stations in the brain. They're forever relayed. And I was wondering, when do they ever get processed, right? So clearly that's a notion that's very limited. The idea of encoding and decoding is one step more sophisticated than that, but still limited. And so the important thing is to see, what can that buy us and what are the limits? What it buys us, I think, is a perspective on what is the transformation from the sensory evidence, from the signals at the sensory surface, to the activity patterns in the brain. And that is the encoding. And then when we take that perspective and we want to say, well, this serves some function, then we have to assume that somehow it's read out, and then we view the rest of the brain as the decoder. But of course, where the encoder ends and the decoder begins is in the eye of the beholder. It depends on the region X that we chose to focus on, right? And if we move on from X to Y, then the little piece of processing from X to Y that was part of the decoder is now part of the encoder, right? So that is one thing to keep in mind. And another thing to keep in mind is that the decoder is not the inverse of the encoder, right? So when often we think of encoders and decoders as inverses, but that doesn't make sense, because then this begs the question why you don't just copy the input. Why would you? And so, we have to think about what is the purpose of the encoding. And there's a number of possible answers. You might want to compress it, for example, for passage through a limited channel like the optic nerve. You might want to transmit it through time. You might want to change the format so that other inferences are computationally close or can be sort of reached in few steps.
Yeah, so Paul, I think so I agree with you that you can look at brain processes as encoding and decoding, and that might be a useful perspective. But at least for one half of the Onion paper, and this might also underlie your initial confusion, we are talking not about encoding and decoding in terms of what the brain does, is about what we as researchers do.
Right.
Okay. So this is the first onion that you have, and that's the data onion in a way. And this is the one that I feel most comfortable with, because, that's just my view as a statistician on the world and on the things that I'm doing. And there, it doesn't matter what the brain does. It's the thing is, like, I want to learn something about what things happen in the brain and what's the relationship to things that happen in the world. I want to study representations. And I can either take an encoding or decoding view, and people do, right? And this is something that Jack Gallen in his lab has talked a lot about you can either say, take an encoding view, meaning like we're going to explain brain activity and predict brain activity from the features in the world, or we do it the other way around. We pretend that we don't know what happened in the world, and we read out or predict the things that happen in the world from the brain activity. And that's the decoding field.
That's how decoding is mostly used, I think, in the literature. Right?
Yeah, and I think that is still the first perspective that comes to mind when you hear the term, and that's also the one that I think is least confusing and makes sense. So it's not about what the brain does in this case. It's about how we treat the data that we get and how we try to make inferences. And so there are these different styles. Now, the interesting thing is that it doesn't matter. You can do encoding and decoding models in visual cortex, so an area that in your perspective takes information in. You can also do encoding and decoding models in motor cortex, where you think like, oh, why do you do an encoding in motor cortex? Motor cortex doesn't encode anything. That's not the point at all, right? It's the type of statistical tool we apply to build this relationship. And I think what's nice about the methods or the class of methods that Niko and I have worked on is that it acknowledges this is a subjective directionality that you choose, and there's no end of discussion in the literature about whether you should do encoding or decoding, blah, blah, blah. It's kind of boring from a statistic point of view because ultimately it doesn't matter, right? It really doesn't matter whether which direction you do, you're going to get similar results and people are confused. And so RSA and PCM lie right in the middle. They say it's neither encoding or decoding. What we have is two complex multivariate patterns. We want to find the relationship between them. And can we build models of that relationship? And that's what really kind of is the main thing. So I think the surface discussion, should you do, what type of analysis should you run on your data and so on, is kind of misled in sometimes that ultimately, let's just get clear what we're trying to infer here.
Well, that's what the representation models do, is just tease out the information that is in the activity, in the population that's represented. I don't know, maybe we should step back and, well, we could either start peeling the onion, or talk about what a representation model is first. I'm not sure what the best way in is.
Maybe we should slightly motivate, because,
Sure.
what does this all about? So really what it's all about is that neuroscience needs to decide how it's going to evaluate computational models. So we're getting these richer and richer measurements of brain activity, tens of thousands of channels. This is always advancing in humans and in animals. And that's wonderful, and it's inspiring, and it gives rise to this vision that we can just measure everything and build it up. But we also need the top-down approach, where we're building these models. And the models are also getting very big, and now we can simulate many aspects of cognition at scale, so at the complexity of the information processing that's actually going on in the brain to some extent, in some sort of subcategories like vision, or certain aspects of motor control and certain aspects of higher level tasks. But then as we build these models, we also have to be able to test these models with brain activity data. And this is a problem that is not getting enough attention. And Jörn and I have been thinking about this for a long time. And I think it's great that sort of more people are getting interested in this problem. But there are really fundamental questions there. And these are both fundamental questions of theoretical neuroscience and sort of more methodological questions of, okay, here you've got your dozen neural network models, and you've got your brain activity data set from array recordings. So now how do you adjudicate between these models, right? And it requires some difficult decisions, because you have to decide what aspects of these high-dimensional measured data do you want to rely on for making that comparison. So you could naively think, well, let's have the models predict the brain activity, right? But then how are you going to do this? Are you going to predict every single neuron, every single spike? That seems too much of an ask, because brains are idiosyncratic. Here we have three brains that, at the level at which I'm interested in brain information processing, are identical in their functionality, but in terms of their particular neurons, they're all unique. And so we have to abstract from these idiosyncrasies somehow, right? And then in neuroscience, there's been sort of these long traditions of somewhat separate literatures discarding different portions of the measured information. And the goal of the Onion paper was to kind of systematize this range of approaches that exist. It's really kind of a review that brings the range of existing approaches to what to discard and what to focus on when analyzing brain activity and using brain activity measurements to adjudicate among models, to bring that into a framework where we can understand how the approaches relate to each other.
I think that one of the most valuable things about this approach, and this is some sort of deficit that we have as humans, I think, because even when there were single neuron recordings, maybe a few people thought, hey, maybe, one day we'll be able to record 2 neurons at a time. And then, it gets bigger and bigger and bigger. But what has been lacking is what happens when we get there, when we can record every single neuron, at the sub-millisecond timescale. What do we do with it? And as humans, we have this tendency to think, well, we'll figure it out when we get there. So I think that this representational model work and just conceiving of it as an onion, as the whole, is really important work.
Yeah, I think, for this part of the paper, it's really important. What we wanted to achieve with the paper is that to make explicit the often implicit decisions that are being made by saying, like, I'm going to analyze my data like this, right? People usually don't take a justification of why they exactly do decoding or why they just look at a map, a somatotopic map of S1. Why do they choose to present the data in that way or analyze or think about the data this way? And these are often implicit decisions, and they have consequences. They have consequences on how the authors think about the paper and influence the way theories are built. So just to give an example, as we all know, neuroscientists love maps. And one of the iconic maps we have in all our textbooks is the homunculus in primary motor cortex.
Yeah.
Right? The idea that there are these areas that relate in a one-to-one relationship to body parts. Okay? And this is very deeply ingrained in people's brains now, because it's just such a tempting and conducive kind of like, you look at the map and you just understand it. So this is kind of what, in our way, is kind of like, this is basically looking at the outer shell of the onion, right? How things are arranged on the cortex. And now I think the key insight came in, like, studying figure representations in M1 and just, staring at these maps, knowing that we measured them highly reliably, and just looking at that intersubject variability where none of them looked like the homunculus. They look all different and they are overlapping and messy. And you can generate data analysis pipelines that give you, like, you can rescue that somatotopic map and it's there and you can talk about it. But you're missing, you're missing the main point, right? You're missing the main point that there's some structure there that relates to how we move the hand that has nothing to do with the somatotopic map, that you cannot understand when you just map in a winner-take-all fashion. And so that's kind of the key idea, that we need to dig in the onion. And now the question is, okay, if we don't look at maps and we want to analyze population codes in a more general fashion, there are many ways of doing this. And which feature are we tossing everything away and just saying, can I decode this physical feature from there? Do I look at the covariance matrix? Do I do PCA? Do I do more fancy methods? Do I take into account where those neurons are on the cortical sheet at all? These are all decisions. And looking ahead, I think people will make these decisions, and that will influence the way they conclude in their papers and the way they develop their theories. And it's really important to understand that the tools we use shape the way we think. And that's kind of the way that the first half of the paper really is targeted at this data analysis process.
Starting at the outside of the onion, then, should we just start there and peel it?
Yep.
All right.
So the data onion, you mean?
The data onion, yeah. And then we'll rebuild it to the scientist. Yeah. Well, we can go either way. I don't know. We have...
Okay. Let's do the data onion first really quick, because that's easy.
Yep.
There's only one onion.
Okay.
There's one onion,
See,
yes.
we disagree on everything, but, that's...
Yeah. There's an onion that you cut, and then there's super glue to put it back together. So
that's the core of the onion. Let's start with the whole onion.
Yeah.
All right. So someone's in an fMRI machine, and you get data about the homunculus and when they move their thumb, where it lights up. And so you have the location and the activity of the cortex. And or, you're recording single neurons or, EEG or whatever. You have these massive recording techniques, and you're getting location information and activity information. Is that all there is to say about the outside of the onion?
Well, yeah, no, I mean, that's the data that goes. That's a whole onion.
Yep.
But now what do you do with it? And there's one tempting thing to do is just to label each spot on the cortex for which body part it responded most. And ta-da-ta-ta, you get a homunculus. Kind of. A little bit messy, but, it's there. Right. It exists, right? Now, the tempting thing is that you then stop thinking.
Envision it would be retinotopic mapping and higher up, finding the category selective regions in the ventral stream. And
the insight from our own work is that when you do that, it makes people very happy. It makes reviewers happy. It makes readers happy. It makes everybody happy.
Television programs?
It's just, you have a map. You understand what the thing is doing. And the treacherous thing here is that you are only looking at the outside of the onion. And really, how things are arranged on the cortical sheet is probably not that important. For the computation.
You could argue either way. I think, these things are very replicable and they are true. And I think the placement of components in the brain is very important. And all of this literature is going to be a treasure trove in decades to come for building models that take the spatial relationships into account. For example, things that are closer together can interact at shorter latency and with shorter connections and less metabolic costs. So this is all totally key. However, just mapping it out doesn't mean that you understand the mechanism, right? You haven't understood how the information is represented, what format it's represented in, and how the computations work, right? So it's sort of a first step. It's essential, but it's not the whole story.
Yeah, you guys don't burn your architectural bridges. I'll be talking to Danielle Bassett in a few days here. So she has something to say about architecture. All right.
So then you can take the radical other view and saying, and that's basically in the original Haxby paper, that's kind of this one. Let's not think about FFA and fusiform face area and parahippocampal place area. Let's just think about the information that we can gather from it. And that's like, basically removing any information and just using a linear classifier or even correlation to decode something. Is there information, linearly decodable information in this data?
So you could scramble all the neurons in an area and you could decode all the same information, right? And at a finer scale, actually, it is more random. So does it matter exactly where those neurons are in the brain to computational function? And this is not just in imaging, but also in electrophysiology. As people got into array recordings and looked at populations of neurons, they became interested in not only thinking of the codes as population codes, that has a longer history, but also analyzing them multivariately as population codes. And so there's this whole separate literature where you say, let's not care about where these neurons are, let's care about what information they convey. For downstream.
And even, going one step further, it's not only forget about where they are, also forget about tuning functions of individual neurons, because it's only the population now that is important. And not only the population, and this is where I think statistical concepts are really important. When you look at a method like linear decoding or correlation, you can actually, it doesn't use the whole data. So there's this concept in statistics that's called the sufficient statistic. So if you get a result, you base it on data, but there's this intermediate point, things that you can compute on the data that is sufficient to get exactly that result. And when you prove what a sufficient statistics is for a certain method, you know exactly what information is discussed. And so this leads us then, if you just look at linear decoding, the sufficient statistics for linear decoding is the covariance matrix of the data, or more generally, the second moment matrix of the data. And it forgets everything else. So whether it's like the distribution, whether there are special clusters in your population, all of that is discarded. And you're only interested in that one statistics that you choose. And so I think thinking about very critically with each method, which parts of the data is it actually looking at. It looks at all the neurons, it looks at population, but you can actually generate other populations that if you would show these populations to a neurophysiologist, you would immediately say, no, this is not a real neuronal recording. Neurons don't look like this. And it would have exactly the same second moment. And the decoding would be exactly the same as the real data. So if you choose to use this tool, you wouldn't be able to distinguish it, right? But there are features in the data that are just not in this, that can't be captured with this method. And so looking at one feature at a time, can you decode this feature or this feature? That's kind of the feature fallacy that you just, basically look at it. And now representational similarity analysis and pattern component analysis go one step further in just saying, we're not interested in a specific feature. We don't even know that there are specific features. We don't want to, we don't necessarily want to think about features and what feature, which neurons encode which feature. But we're just going to think about the structure of the neural activity and the structure of the feature space and relate it to each other. And so this is basically now you haven't gone further much in terms of finer arguments about the distribution, or you're still throwing away the spatial information. But now you're looking at all features all possible features that you could think about in one go. Right? And that's a much more complete description of that particular view.
So this is going from the stimulus response matrix, where we have all the stimuli along the vertical and all the responses, all the neurons along the horizontal, and an activity in every entry of that matrix, to this square matrix that's stimuli by stimuli. And this could be the second moment of the stimulus response matrix, or it could be the distance matrix. And these two are actually equivalent. You can compute one from the other. And you can think of this sufficient statistic as characterizing the representational space or the representational geometry. And that's most intuitive to me when I think of the distance matrix, where every point you have for every stimulus, you have a response pattern that's a point in the multivariate response space, and then you have the distances between these points that define the geometry of the ensemble. And that's what's defined by this intermediate level summary statistic that we found particularly useful.
Just for clarity, what does a second moment measure?
Oh, in a way, it's the variance covariance matrix of the data. Okay. And in general, we say second moment because it's not just variance and covariance. So basically, how similar is this pattern? How much does this pattern correlate with this pattern? Mm-hmm. But we also take into account that one pattern might be much brighter than the other one or much more active. Okay. In one condition, the brain is very, very active. And in one condition, the brain isn't very active. If you just take the covariance across neurons or voxels, you would subtract out the mean. So second moment is just a statistical concept for a covariance matrix where you didn't subtract out the mean, basically.
Okay. Very good. So we've peeled away one level of the onion. We have these representation models, pattern component modeling, representational similarity analysis. I'm not sure if we've said enough about them.
Oh, we're going the other direction, right? So what we did, we looked at the outside of the data onion. We looked at the outside of the data onion. We went all the way in the center and say, okay, if you only look at one feature, you're just looking at this. It's like one kernel, and you might not find the center of the onion. Now, the level that we've described and that our research is mostly focused on is using the second moment. And that is already a very rich description of the neural population code.
And just to be clear, those linear decoders, the center of the onion, there's lots of tiny little bulbs in there, lots of linear decoders. There's no one center like you just...
Well, because some people tries to decode velocity and another person tries to decode muscle activity, and another person tries to decode position or torque or snap, and they all can declare victory because they all can decode that particular feature from the thing. Mm-hmm. And not all of them are just getting one kind of needle in the haystack view of the whole representation. And I think what we're pointing out here in the paper is that if you really want to describe the whole representation in the constraint that you only look at these things that you could read out linearly, features, then you need to look and
put them all together. Yeah,
you need to, and that's what representational similarity analysis does for you. Now we still have thrown, and so now we're going out. Now we're saying, what did we actually peel away to arrive at this level? And in many ways, I still think this level is a very happy medium between peeling away and not peeling too much away. And I think, we will keep doing things on this level, but we want to acknowledge that we peel things away, right? We throw away all the spatial information. Mm-hmm. And it's, we in our lab and many other people are looking at the spatial information kind of through the new lens now and saying, like, we don't want to just do like maps, but we want to acknowledge that the spatial information is very variable across subjects. And the exact XYZ location might not be that important, but the relationship between how these things that we can describe as representational models and how they map on the surface, that's something.
Do you think of it more as as a constraint in a principle of information coding, or is it part of the information coding itself?
Well, it's a product of learning, right? So there are learning rules in the brain. There is architecture, genetically predetermined architecture, but there is also the learning algorithm that is important in the census system. And that learning algorithm learns on some data, and it has effects. Now, that learning algorithm isn't spatially blind. It's, nearby neurons have more, more opportunity to communicate. They might build more long-term potentiation with each other. So there are things that could be changed, but actually where the learning algorithm follows that gradient basically and makes it so. And looking at when we're at the level of these relatively fine-grained activity patterns that we look at, maybe within a cortical region, I think they are the product of some learning and some, which is partly a random biological process, but there's some commonalities about how these look like when you look at them on the cortical surface. It's going to tell us a lot about what they're trying to optimize and what they're trying to do and what computational function they serve. Okay, so rather than thinking about them as constraint, they're the product of some biological constraints that are genetic and some learning rule that was applied to things that the system tried to do when it grew up.
How things were learned, in what order, at what time, those sorts of environmental considerations.
Exactly. Yeah.
That's interesting.
And what the system tried to optimize, what was important for that brain region to do, right? And I think understanding those things, ultimately we need to come up with theories that link from the things that the brain wants to do. And one of the hard genetic constraints, but I think the literature on plasticity, for people who lost their arm or born without a hand or are born blind, tells us that there's a lot of stuff that happens or can happen when the system just, misses one input. Right. And the representations might look very different, and understanding those features are important.
This also maps well onto your work on sequential motor activity and how that might get encoded within a spatial distribution, right? Depending on how you learn it and different types of sequences, which we can't really get into, but it's really interesting stuff.
Yeah, it's also, relates a lot to work that my colleague Tamar Markin at UCL does on looking at amputees, right? So what happens if you lost a hand 20 years ago? What happens to these representations in the cortex? What happens when you never were born with a hand? And so I think that this is a really kind of interesting field to look at about how do these activity maps come about. So now we're not saying it's the exact map that is important, but there is the spatial relationship between these elements that we've identified by analyzing the second moment. That is still important, and coming up with that is I want to keep an open mind. It might turn out to be really, really informative. It might turn out to be very simple, that there's just a smoothness constraint on the cortex and, whatever, and that there isn't really much interesting things going on. But it's clearly a data feature that we haven't looked at and that we might want to feel uncomfortable about discarding so easily.
Niko, do you want to respond to that? And also, I see that we're 2 minutes until the half hour.
Maybe just to wrap up the onion part, maybe good to summarize a little bit, right? So the whole of the onion is the stimulus response matrix for some little area in the brain that you've recorded from with an array or with functional imaging. And you've presented a number of stimuli, and you've got a response for each of your response channels, each of your neurons. And then you also know where these neurons are, right? And then some neuroscientists want to make a map out of that. They want to make a little map of that patch of cortex and show the cell activities. And then others say, well, I'm really interested in computation. I want to see what information is in there. So I am not so interested in this map. So I'm going to throw away the locations matrix and just look at the activity profiles. And then I have some summary of the distribution of activity profiles. And that's the level at which Jörn has defined representational models and as a probability distribution over activity profiles, which I think is a extremely important concept, and to me an inspiring concept, because this keeps more information than I want to keep. I want to go one step further, or where I say, well, not only do I not care about the exact positions of those neurons, I also don't care about the distribution of activity profiles. I care about the geometry of the representation. And that can be characterized by the second moment or by the distance matrix. And interestingly, this sufficient statistic now gives you, under certain assumptions about the noise, for example, let's say it's a Euclidean distance matrix and the noise is isotropic. In that case, this gives you all the information that is encoded about the stimuli in the neural responses, and it gives you additionally information about the format of the code. For example, you can then predict what can be linearly decoded, and you don't need the original stimulus response matrix anymore. You don't need the distribution of activity profiles. You just keep this stimulus by stimulus matrix, and that's been very useful to us in comparing human brains to monkey brains and monkey brains to computational model representations. So that is sort of the one layer deeper. And then there are other people who have said historically, well, we don't care about all dimensions of the representational geometry. We just want to check, can this particular important variable be decoded? And that's where linear decoding is, right? This is people who are interested in asking whether particular information is not only present in the data, but is present in a format that can be linearly decoded. And they're interested in that because they're saying, well, if it can be linearly decoded by us, then it can certainly also be read out by downstream neurons, right? And then you could go further and you could say, well, maybe we should restrict this more and say it should be decodable from a local set of neurons. And then you have sort of more restricted definitions of the decodable information, and that's where you get into the core of the onion. And in the end, you're back with looking just at a single cell and the cell activity of a single cell. And of course, that was the classical approach in neurophysiology, where you only had one cell to look at, and you tried to understand the cell activity and the tuning properties of a single cell.
Or on a good day, you have two cells on one electrode, but that was a good day. Okay, then that's a great summary for peeling the onion. So let's put it back together, and then I guess we'll wrap up pretty quickly.
So the classical studies in neurophysiology that looked at cell activity were interested in single neurons. Now you can ask why. On the one hand, that's a place to start, and the idea being you look at one cell at a time, and then once you've looked at 100 billion of them, maybe you understand the brain, right? But of course that's a naive hope, and everyone knew that. And even from studying single cell responses, people understood that the code isn't fundamentally a single cell code, that in order to interpret what these findings mean, they had to think about the population of neurons and the information encoded in the population. And so the concept of the population code was very strong in theoretical neuroscience before it was strong in the analysis of data, right? So people started caring about what the information contained in the population and the format at the level of linear decodability, which is not the only biologically plausible concept of decoding or reading out a brain representation, but it's an influential one.
It's almost the easiest, intuitive way to do it, I would imagine. Yeah.
And well, so partly it's that you have a hammer, and then, it looks very tempting to just pound that nail.
And now you have this better hammer of, measuring lots of cells, and you have the linear decoders, and you can ask these more specific questions. But then, of course, you want to say, well, we decode certain dimensions. Why don't we just decode all dimensions? Why don't we just look at all readout dimensions? And that leads you to the representational geometry and the second moment. All
within a certain range of brain area and stimuli.
Yes. Yeah. And so then you're at the representational geometry. So you could say, well, maybe we're happy there, right? I mean, Jörn and I have certainly been happy there. Why would anyone want to add further outer layers to the onion? But there are good reasons. So you could say, well, now all you care about is what's linearly decodable or the representational geometry, but the same representational geometry can be spanned by many different sets of single neurons with different kinds of tuning. And not all of these might be equally easily computed from the stimulus input. So it might not be irrelevant which particular set of a single neuron tuning characteristics spans this representational space. It might be important for understanding the computational function, and that's where we might care again about the particular encoding and look at the whole distribution of encodings, right? And then if we're at that stage, then we can say, well, part of these computations going on is lateral interactions between these neurons. It's a recurrent neural network. These neurons are talking to each other, and that informs their tuning, and who they can talk to depends on where they are. They can't talk at equal cost to a neuron that's far away. So the placement is important, and then we're back with the entire onion.
So we're kind of back at maps, but the key lesson from the journey, I think, is that the way we look at these maps needs to fundamentally change. And I think that's from a statistical point of view, we don't really have the tools to really analyze those types of maps very well. And that's I think, a kind of interesting frontier. And trying to ask questions, it doesn't matter.
Doesn't matter, yeah.
Is this, doesn't matter at all. Is there something to be learned from? Where are the regularities? And that's I think, is really kind of interesting things to pursue.
And one of the things that, and you can correct me if I'm wrong, but that I'm not seeing in the onion as a whole at any level is the actual dynamics, how things change over time, because all of these measurements are made within a window of time and then averaged or, whatever.
Yeah, I think this is a particular lens that we've applied here, which is very driven by the fact that we both started with fMRI, functional magnetic resonance imaging, where you just don't have that information.
Yep.
So it's like having only a single cell. You don't think about populations right away. And in this case, it's just a constraint on the measurement technique that at the timescales that we might be interested in, it doesn't deliver very reliable information.
Yeah.
Now, yes, that is absolutely another dimension, and we can write another onion paper about it. So
growing the
somewhere, some other onion that the temporal information is. But we didn't focus on that.
You can animate all of these sufficient statistics and look at them as movies, right? You can have the representational distance matrix as a movie, or the second moment as a movie, or you could have spatiotemporal patterns and then do the same analyses. So there is some sort of straightforward ways to extend it, but it's also true that there are some fundamental questions that are in some ways analogous to the questions that we focus on in the paper, which is that they're about correspondency between different systems. And our paper focuses on spatial correspondency and how we can overcome the idiosyncrasies in terms of spatial layout of things at a sort of fine scale. And then when you get into dynamics, there's also the problem of temporal correspondency, how do you integrate over time? How, maybe one participant gets the joke slightly later than another one, and but still fundamentally their brains are going through the same cognitive process and understanding the movie or the joke. And how can you relate these two processes to each other?
Guys, I think that this is a good place to leave it with the onion, and I'll point people to the paper as well, and I'll make sure that people remember when you're reading the text, look at the right figure, and vice versa. It's hard to understand if you don't do that. Maybe just quick hits here. Do spikes matter? Does spiking matter?
I think spiking is absolutely essential for neurons to signal over long distances. And it might also be computationally important, but I don't know about that yet. And it's not on top of my list of biological details to add to the models that we're building.
To the neural networks.
That's subjective. So it could be essential. It could be an essential feature.
It's pretty likely with high probability that there exists some level at which spiking isn't important anymore.
At which it is not important.
Yeah, when it's not important, right? So that I were told. I think the proof is in the pudding. What the real importance of spiking versus continuous rate codes are, and I don't know.
Finally, Jörn, what's something that you used to believe that you now think is naive?
Oh, lots of things. I forgot them all, I think. This is this is what the brain does when you believed something that was naive, and then just to maintain your self-worth. And
maybe you used to believe that something was real, and now you believe nothing is real.
No, I didn't. I don't believe that nothing is real.
All right, I got you. I have that quote as well. What about you, Niko?
I think when I started, I thought it was kind of glamorous to try to find out how the brain works, but also impossible.
Yeah.
And I thought anyone who thinks that it's possible to succeed must be naive. Now I think, I've kind of lost this confidence that it's impossible. I think, who knows? Maybe we have a shot.
That's the directly opposite way most people go into it. You have to start off naive and confident that you can figure it out, and then it beats you down over time, right?
Maybe I'm just getting more naive as I get older.
Well, the problem is if we figured it out, we're out of a job, right? So,
yeah,
it wouldn't be a good thing to do.
Yeah, slow it down a little bit. Well,
I'm not worried.
Yeah. Thanks, guys, for being here with me, and I appreciate you both being on. It's been fun.
Okay.
Great. Good to see you, Paul.
Good to see you, Niko.
Bye, Paul.
See you, Jörn.
Bye, Niko. Okay, talk soon. Bye-bye.
Brain Inspired is a production of me and you. You can support the show through Patreon for a microscopic $2 or $4 per month. Go to braininspired.co and find the red Patreon button there. Your contribution will help sustain and improve the show and prohibit any annoying advertisements like you hear on other shows. To get in touch with me, email paul@braininspired.co. The music you hear is by The New Year. Find them at thenewyear.net. Thanks for your support. See you next time.